This will be a fun chapter. So, chapter 3, we talked about prokaryotes, and in this chapter, we are going to talk about eukaryotes. So, eukaryotes have been around for about 2 billion years. That might be 1 or 2 years off. Who knows? Both prokaryotes and eukaryotes evolved from what they call the last common ancestor, and then you remember that the archaea branched off of the eukaryotic line, right? So whatever this supposed last common ancestor was neither prokaryotic nor eukaryotic, but apparently um, must have had something in common with both, right? Because it gave rise to both. Um, there is a concept called endosymbiosis. Endo means within, bio means life, and sim means together. So what this is a relationship among organisms, and we'll talk about some examples later on. But they use this as a possible explanation for why eukaryotes have membrane-bound organelles. What they think may have happened was some pre-eukaryotic cell engulfed some prokaryotic cell, and rather than digesting and eating the thing, it ended up living inside of it. And then when the cell divided to multiply itself, it apparently divided the other cell along with it and somehow became one organism rather than two. Okay, so that might be a stretch for some of you, um, but I don't really know. It doesn't really matter at this point because we certainly know it does. We do have eukaryotes with membrane-bound organelles. But anyway, this kind of shows a picture of it and how they think. Pretty much explains just what I said. So this is showing this cell that engulfs some other prokaryotic cell, and I'm going to tell you a little bit too why. Um, uh, well, I may forget, so I'll tell you now why they think that this might be the case. Um, is because when they look at the DNA of a mitochondrion, there are sequences in this mitochondrion. I think we do talk about this. I'm probably getting ahead of myself. The sequences of this mitochondrion have bacterial sequences in them that are only found in bacteria, and that's sort of a really good argument as to why they think this came from a bacterium. Um, anyway, so this larger cell engulfs the other one. This one then can divide inside of it, and this is what they're trying to show is how a mitochondria may have started. Um, and so we end up with several of these in here, and then the next thing you know, they're living together, and we end up then splitting to animal, fungi, protozoa, and then algae and plants over here, and all the animals and plants and whatnot that we sort of know of today. So that's the endosymbiosis theory. So the first eukaryotes were most likely single-celled. I mean, I can't imagine how they would be multicellular organisms without being single-cell organisms first, so that's probably true. They think that they then began to aggregate and form colonies, and we know they form colonies now, so that's not really much of a stretch. What they think, though, is cells within the colony became more and more specialized until they were actually a completely unique kind of cell that couldn't live without the other cells around it. And the next thing you know, 10 billion years later, or I guess million, let's say, uh, they become an actual separate, unique organism that is multicellular. Mainly the disease-causing eukaryotes are what we're going to talk about in this chapter, such as protozoa and fungi and helminths. Bearing in mind, I may mention animals, humans, things like that as we're discussing this, only because I want to bring up the fact that you have learned some of this already, and I just want you to make the associations, okay? Um, oh, so we are going to talk about this. Okay, so what's the evidence of this endosymbiosis thing? Mitochondria of the eukaryotic cells resembles a prokaryotic cells, and they contain circular DNA, just like prokaryotes, and they are capable of independent division, which makes them almost seem like they're a separate organism. And they contain prokaryotic ribosomes, which were the 70S ribosomes, unlike the eukaryotic cell, which has 80S ribosomes. Their membranes are like bacterial membranes that are also inhibited by drugs that only affect bacteria. And I swear this was in here, but as I said, the DNA sequences are also only seen in bacteria. They also think that chloroplasts, which are in photosynthesizing organisms such as plants and algae, developed from a photosynthetic bacteria called a cyanobacteria and led to these chloroplasts. So which of the following characteristics is evidence that mitochondria were once prokaryotic cells? And circular chromosomes, yes. Prokaryotic ribosomes, yes. Capable of independent division, yes. Membranes that are inhibited by antibiotics, yes. So the answer is everything. So most eukaryotic cells have the things that we're going to list right here. Cell membrane, a nucleus where the DNA is, mitochondria that create ATP, endoplasmic reticulum, Golgi apparatus, vacuoles, cytoskeleton. Uh, Only microbes have a glycocalyx. You're not going to see a glycocalyx in multicellular organisms. And don't panic, we're going to talk about every single one of those things in detail. So let's diagram out with this fancy flowchart here. Eukaryotic cell. So on the outside, they might have appendages, okay, which would be in the form of flagella and cilia. Microorganisms might have a glycocalyx, which could form a capsule or a slime. This should be familiar to you. The outside of the cell is definitely going to have a cell membrane, and it might have a cell wall. They don't all. Inside the cell, they're all going to have a cytoplasm. They're all going to have a nucleus, which is going to have a nuclear envelope around it, a nucleolus, which is just a ball of proteins and chromosomes, and of course, they've got to have chromosomes in them. Organelles such as endoplasmic reticulum, ribosomes, Golgi apparatus, lysosomes, mitochondria, chloroplasts. They're all going to have ribosomes because they've got to make protein, and they're all going to have some sort of a cytoskeleton made of these microtubules, intermediate filaments, or actin filaments. Okay, so this picture here, which is figure 4.2 in your book, is what you are going to want to reference as we discuss this stuff. By the way, this right here is figure 3.1 from the previous chapter, and I think what they're trying to show is an average size comparison. But bear in mind, these things are all different sizes, so probably not all that useful to have that there. But that's a prokaryote down there. Okay, so let's just quickly do an overview of what this stuff is. So this right here is a lysosome, just a little membrane-bound organelle that would have in it lysozymes. A Golgi apparatus, the stacked orange thing right here, mitochondria, and this is this double-layered structure right here. Intermediate filaments, microtubules, and actin filaments, all of these things up here are what create the cytoskeleton. The cell membrane here, nuclear membrane, which is this membrane around the nucleus. And then, of course, this whole structure here is the nucleus. Inside this ball is the nucleolus, which is made out of DNA. This structure all around the nucleus here, this all these folds, this is the rough ER, and the reason why we call it rough is because it has these ribosomes on the surface. Smooth ER over here does not have ribosomes on the surface. Hard to see here because I messed it up, but the flagellum, chloroplast, centriole, cell wall, and glycocalyx, we'll go through all that as we get to this, okay? So here's the picture of that same cell. So let's talk about flagella. Flagella are just like in the prokaryotes, used for motility, which is movement through space, right? So it allows these organisms, just like with prokaryotes, to move toward things they want and away from things that they don't want. Okay, the name we use for that is locomotion. And you will see this locomotion either because of cilia or flagella in protozoa, algae, and some fungal and animal cells. Animal cells, example, the obvious one would be the sperm, right? So there's differences between the flagella of eukaryotes and prokaryotes. And remember, the prokaryotic flagella twisted and or spun around like a propeller. Eukaryotic flagella doesn't do that, okay? It's also a lot thicker. It's covered by an extension of the membrane of the cell. So rather than sticking through the cell, so to speak, it's sort of, or through the membrane, I mean, it sort of comes out and is still covered with the membrane, sort of, okay? So it's a long structure. Um, it's got a sheath around it, as we just mentioned, and it's actually made up of these microtubules in a 9+2 arrangement. And so if you look over here at the picture on the right, there are 9 microtubules on the outside and 2 microtubules on the inside. All right, so that's where we get this 9+2 arrangement. And so the way these things move is there's interactions, kind of like actin and myosin in the sliding filament mechanism of a sarcomere, how they interact with each other. Sort of what these things do. So these middle fibers will interact with the outer fibers and create a whipping motion, okay, not a spinning motion. And of course, this is going to require energy, right? Cilia, similar structure to the flagella, but they're a lot shorter and there are a lot more of them. Now in an animal, as far as I know, the only one that has a flagellum is a sperm, and it only has one. But some of these can have more than one. But when it comes to cilia, they can have hundreds or maybe even more. Um, so cilia are found in only one group of protozoa, and lots of animal cells have them, but they're not, as far as I know, in animal cells, they're never a free living cell. They're always attached cells, such as your respiratory tract, fallopian tubes, the ependymal cells of the ventricles of your brain, things like that. So they occur over the whole cell surface, except in these animal cells, they're only on the free surface. They beat together. I describe this to my students as sort of like, at least in animal cells, kind of like seaweed moving in the waves, but they move pretty quickly in these protozoa. But they sort of act like oars in a rowboat to help these things move through space. They can also, though, function as a feeding mechanism um, or filtering mechanism. So the glycocalyx, remember talking about in the prokaryotes. Now remember, only microbes of eukaryotes have these. These are not in multicellular animals or organisms. Okay, it's also known as the extracellular matrix in these. And it's the outermost layer. This is outside of the plasma membrane, of course. And so this is what is actually in contact with the environment. So usually composed of polysaccharides, just like the glycocalyx in the prokaryotes, um, and appears as a network of fibers or a slime layer or a capsule. So same thing, the slime layer is not as tough or in is close contact with the cell as the capsule is. So pretty much for the same reasons we have it in the eukaryotes. Protection helps it adhere to the cells of helps the cells adhere to other surfaces like glass and things like that, and can be used as receptors to gain information from other cells or about the environment. Okay, the cell wall. Animals, protozoa, and helminths do not have a cell wall. Okay, we don't have one. Um, so we only see these in fungi and plants. So it's rigid and provides structural support and shape. It makes them very, very tough. Um, Different in chemical composition from the prokaryotic cell walls because it's a thick layer of polysaccharide, in other words, sugars. Um, Literally, the cell wall of a plant is sugar. I mean, wood that you burn in your fireplace is sugar. It's just a type of sugar we don't have access to because we don't have the enzymes to break it down, um, which we call cellulose, by the way, or fiber. When you eat it and can't digest it, we call it fiber, but it's all the same thing. In fungi, it's called chitin, which is also a polysaccharide. Um, can also be mixed glycans, by the way. So this right here shows a picture of a fungal cell wall. So here's the inside, here's the outside. So notice this made of chitin, and we have this glycoprotein and these mixed glycans that can be on the outside of this. And also it may have, if it's a single-celled organism like a yeast, this glycocalyx layer on the outside. So the cell membrane, also known as the plasma membrane, I should have written that there, maybe make a note of that somewhere. Um, Phospholipid bilayer with protein molecules and other things in it, okay? They also contain sterols of different kinds like cholesterol to stabilize the membrane. Very important to have these sterols in the cells that don't have a cell wall because otherwise they would just fall apart. And as I've mentioned, it forms a selectively permeable or semi-permeable barrier, which lets certain things in and does not let certain things in and out, for that matter. All right, so let's just go through some of the organelles of these cells. This thing right here is a nucleus, all right? Keep referring back to that figure 4.2 if you need to see where this fits, but you can see we're kind of pulling this out of this cell right here. So this is a microscopic view of the nucleus. So right here is the nuclear membrane, and all of this out here is the endoplasmic reticulum. So the outside of this thing, what creates the boundary of the nucleus is the nuclear envelope or nuclear membrane. Uh, I wish we didn't use all these different terms and words for things, but unfortunately that's just the way it goes. A lot of this stuff develops in completely different parts of the world, and so they sort of become so commonplace wherever they are that they just keep them, and you just kind of have to learn more than one term for things. The other thing that happens in science is a lot of times a term will be coined or whatever, and then sometime later there's somebody down the road decides it's not as good of a term, and so they come up with a new term. And until that gets replaced, you need to just know both of them. So anyway, I'm just the messenger. Um, so there are two membranes separated by a space. So this is just another phospholipid bilayer, right? Um, Through it, though, we have these little pores. And this is much more porous, by the way, than like the cell membrane, but it really is fairly similar in structure to the cell membrane. We don't have the same receptors, and we don't have the same um, kinds of proteins and oligosaccharides and antigens and things like that on it. But I mean, the main structure of it is pretty much like the plasma membrane. So what we use that for is to get things in and out. And we got giant molecules that need to come in and out of this nucleus like RNA, mRNA, tRNA, rRNA. Those things have to come in and out. And inside of it, we have cytoplasm, so to speak. But since it's in the nucleus, we don't call it that. We call it nucleoplasm. But I mean, it's the same thing. It's coming in and out through those holes, so it's the same thing. Um, Inside of it, we have the nucleolus, which is this little ball right there. This is the site of RNA synthesis. So this is where we're using DNA to make a copy of RNA. It's also the collection area for the ribosomal subunits, the rRNA that we will then send out to make a ribosome. And when you stain a cell, this stains darker because of the higher density of RNA that's in there, and so it takes the stain. Chromatin is these fibers of DNA with these proteins called histone proteins. So the DNA actually wraps itself around these histones. Um, and this is what contains the genes of the organism. Endoplasmic reticulum, that's what that orange arrow is pointing to right there. So these tunnels that we use to transport and store things, and there's two different kinds. The smooth endoplasmic reticulum, which is, I mean, that looks like smooth right there because there's no ERs or ribosomes on it, but it really is rough ER that doesn't have ribosomes on it. So I'm not exactly sure what they're trying to draw there, but it's a closed tubular network that doesn't have ribosomes on it. Go back to that other picture, it shows it a little bit better. We use it for nutrient processing, such as non-protein molecules like lipids. So we package and transport lipids throughout this smooth ER. Rough ER, now that's a good picture there, has ribosomes on the surface. And so when you look at these things under a microscope, these look rough. That's why they call it that. Extends into basically what it does, it creates a passage from the nuclear envelope. I mean, there's a hole that goes directly into the inside of this, and it will actually extend all the way to the cell membrane. So we can transport things all the way through this right out of the cell. As I said, ribosomes are on the surface. And so since ribosomes are on the surface and we use ribosomes to create proteins, then you could probably theorize, and you would be correct, that rough endoplasmic reticulum is for packaging and synthesizing and transporting proteins. So this right here just shows, so that's what we just looked at here. This right here is a microscopic slice of this cell. And this shows these ribosomes with a piece of mRNA attached to it. And what this is right here, this is the protein that we're actually making off of it. What's missing from this picture is the tRNAs. But we'll come back to this a little bit later. The Golgi apparatus is also a place that we modify protein. And we actually can use it to package the proteins and get it out of the cells. For example, hormones in a human, protein hormones. In an endocrine gland could be used this way. Consists of these pancake-looking things that are all stacked on top of each other, and they call those things cisternae. And a cistern is just what is it? It's kind of a collecting area for, I don't know, water and irrigation or something like that. Okay, these, unlike the endoplasmic reticulum, however, don't form continuous networks. These are stacked on each other. It's not all one continuous maze like it is in the ER. They're separated. Always closely associated with the ER, because you can see what this is showing here is an endoplasmic reticulum. They can bud off and actually combine and become part of a Golgi apparatus. And then we will continue to modify whatever the contents are, and then release it again, and release it outside of the cell. So they call those things transitional vesicles. A vesicle is kind of like a vacuole, but it goes the opposite direction. It goes out instead of in. But the vesicle comes over here, and as I said, it buds and then combines with the Golgi. So we may add things like polysaccharides or lipids to these proteins to make a lipoprotein or a sugar protein complex. So these things are called condensing vesicles, so transitional and condensing, and they pinch off of the Golgi and either become a lysosome or go toward a lysosome, or they're transported to the cell membrane and combine with the cell membrane in a process called exocytosis. And that picture doesn't show us a whole lot more besides a microscopic view. So your book talks about these organelles as being nature's assembly line, and that's just because of the chemical reactions that occur from one to another as they process things. So it just tries to talk about kind of how some of this stuff works. So when a piece of DNA is copied um, into a piece of mRNA or RNA of some sort, um, the holes, those nuclear pores through the nuclear envelope, are where those things pass through into the rough ER. Now, once they're in the ER, they can be modified and potentially even transported into one of the Golgi apparati. And once in the Golgi, they can be further modified and packaged into vesicles to be used by the cell, or in the case of multicellular eukaryotes, can be exocytosed out of the cell, uh, like in a hormone or something. And so this just kind of shows these sequences. So we create RNA, and what they're showing that's being created here is rRNA, because this is ribosome parts. So this rRNA exits through the nuclear pore into the rough endoplasmic reticulum. From there, it is packaged and further modified into the Golgi, and then it shows it secreting it via exocytosis. Now, I'm not exactly sure why it's showing rRNA being secreted, um, but that's what they've got shown here. So normally it's not, well, I don't think it's ever going to be rRNA, but some type of RNA, or what I guess what they're probably missing here is the step where we create a protein. So I don't know, it's a little bit confusing, not positive why they drew it that way. A vesicle. So one of the vesicles that we have are these things called lysosomes. So they contain different enzymes. We call these lysozymes. As a matter of fact, one of them is called lysozyme because it's an enzyme that lyses, and you know, lyse means to destroy or to loosen. So we'll just think of it as to destroy. And what it's for is to, well, in the case of white blood cells, it kills bacteria. In the case of all of the rest of our cells, and also these eukaryotic microorganisms, is it is used to uh, recycle damaged cell parts. So there are all kinds of things that cause damage to our cells and cell parts, such as free radical damage, direct radiation damage, heat damage, pH damage, and so on and so forth. And when we get parts that are damaged and no longer useful, they're going to have to be remade. But what do we do with the old parts? We got to get rid of them, otherwise it would just continue to fill up, and that would be a problem. And so we use these lysozymes to be able to break those damaged cell parts down, so we can either reuse the pieces or just get rid of them. A vacuole is a membrane-bound sac that has either fluids or solids, such as large molecules and things like that, that are going to be digested, excreted, stored, whatever. What happens is a piece of the membrane actually pinches off and sort of wraps around the whatever is inside of the vacuole. And so the vacuole, the membrane of it is a phospholipid bilayer, just like the membrane of the cell, because it was the membrane of the cell. Now, generally, we're going to talk about vacuoles as something that's during endocytosis, and vesicles generally would be talked about during exocytosis. So I'm not sure if this is just a mistake here or what, where it says excreted. Anyway, doesn't matter. So they're formed in phagocytic cells, as I mentioned, in response to food or whatever that's being engulfed. Okay. Contents of the vacuole can then be digested once a lysosome actually merges with the vacuole. And so this shows graphical representation basically of what I just said. So this green blob here represents whatever this cell is phagocytizing. So you can see it starts to wrap its membrane around here. It doesn't show it very well. But what happens is once it wraps around, it actually pinches off. So that's why this yellow part is the same color as this, because it used to be it was this section of the cell membrane. Then what happens is a lysosome, which uh, was a vesicle coming off of the Golgi here, merges, and so this membrane of this lysosome actually becomes part of the membrane of this vacuole. Well, once it releases its lysozymes inside of there, you can see how this one big blob becomes several small blobs because the digestive enzymes in that lysosome digest it, and that's how we get from big pieces to small pieces. The mitochondria is where we generate our energy in our cell. Um, Again, remember this is only in eukaryotes, right? So I always call it the powerhouse of the cell. I'm fairly certain I did not coin that term, um, but because this is where we create our energy or generate our energy from, right? Um, Now we will get to this at some point, but just kind of as a pre just to kind of let the cat out of the bag a little early here. We The way we do it, and all eukaryotes, to my knowledge, there's really only, I guess there's really only two things that we use. I'm not going to talk about one of them. Um, it's creatine phosphate, but generally we're going to be concerned with how we break down sugar. So here's what happens. You remember we talked about decomposition reactions and synthesis reactions earlier in this book. But what we do during this process is we take a glucose and we take 6 oxygen molecules and we run them through a series of steps. But essentially what we're doing is we're breaking down the glucose. And when we break that glucose apart, when we break a chemical bond, a covalent bond, it releases energy. And then what we do is we use that energy to create an ATP molecule. We take a phosphate and add it to an ADP to create ATP, and then we can use that ATP sort of like a battery. That's how I describe it. So when we want to do something, we just pop a battery in and use the energy from the battery, and then we go and recharge the battery again in the mitochondria. So what they are, these organelles are round or elongated. We generally draw them as elongated particles or organelles scattered throughout the cytoplasm. So we're looking at 3 of them right here. Composed of smooth, continuous outer membrane, and the inner membrane is all folded up into these things that we call cristae. That's plural for crista. The reason why they're all folded up is because the inner membrane is actually bigger than the outer membrane, and so it would be like stuffing a large balloon inside of a small balloon. Of course, it's going to have to be all wadded up and folded up, right? Um, so all along these membranes are the enzymes and the electron carriers through this thing called the electron transport system that we use for what I've just described to you, which is called aerobic respiration, where we take oxygen, air, AER, um, and glucose and break it apart. Um, so when we break apart the glucose, we use that energy to make an ATP. And it's important that we have these folds, these cristae, because they increase the surface area, and the more surface area we have, the greater the ability we have to produce our ATP. So some interesting things about the mitochondria. When a cell divides, we have to divide all the organelles inside of them, right? Or multiply them because we end up with 2 different cells. Well, what's interesting about these particular organelles is they actually divide on their own as if they are a completely separate organism. And again, going back to that endosymbiosis concept, that's one of the arguments for that as to why people think that that's how uh, eukaryotic organisms with organelles developed. They have circular DNA, just like bacteria, and their ribosomes are 70S, just like prokaryotes as well. And then this picture just shows a microscopic view of the mitochondria. All right, chloroplasts are only found in algae and plant cells. And what they do is they convert the energy from a photon of light into chemical energy. And that process is called photosynthesis. Um, as a byproduct, they produce oxygen, which is wonderful for us because we need oxygen, and our byproduct is carbon dioxide. And so it's the opposite of these photosynthetic organisms. So they resemble mitochondria, but are much larger, and they have pigments in them, usually green, but not always. There can be orange, dark red, purple, but generally they're green, um, and are more varied in shape than the mitochondria are. Ribosomes. So this picture I took from one of those earlier pictures, um, I think showing the nucleus, but there are 3 ribosomes there. So there's all, we've talked about them so much now, you should know what they are, but they're those little dots all over the ER or just floating around in the cytoplasm. And remember, they can be chained into these things called polysomes in some cases, right? So they're distributed throughout the cell. They're also inside of mitochondria and chloroplasts because remember, those essential, I shouldn't say essentially, but sort of are their own little organisms inside of the cell, right? So polyribosomes or polysomes were those chains that I had mentioned. So they're made of 2 parts, a large subunit and a small subunit of what is called ribonucleoprotein, which is actually RNA, rRNA mixed with protein. So as I said, eukaryotic ribosomes are 80S, except in the mitochondria where they're 70S, just like the prokaryotes. And it's a combination of 60S and 40S subunits, which is different from the 70S ribosomes, which were a combination of 30 and 50, as you recall. So what these are is where it says the staging areas for protein synthesis. Really, it's just where we create protein. So what we're showing on this picture here, just to, and we'll go through this in detail later on, but what this is showing here is we hook this particular, this is a piece of mRNA, this yellow thing, and so all 3 of these happen to be hooked to the same mRNA, which means they are all 3 making the exact same protein. It's just that this one is further ahead than the other 2. That's why this one's protein is longer. Once this one reaches this spot, it will be as long as this one, and I think you figure out what I'm talking about. So this is the protein that is being created from the instructions on this piece of mRNA right here. So let's move on to the cytoskeleton. So flexible framework of molecules crisscrossing the cytoplasm, used for anchoring organelles, moving RNA and vesicles. It sort of acts like a little highway, and permitting shape changes, in some cases movement, because it almost acts like a muscular system, sort of, in some of these amoeboid type organisms. But it's made out of several different filaments, including actin filaments. You certainly don't need to be able to, identify which one of these is an actin filament on a picture, on a test, or anything like that. But just be aware there are actin filaments, which are very long, thin protein strands, about 7 nanometers in diameter, found throughout the cell, but mostly inside the cell membrane, or just barely on the inner surface of the cell membrane. Responsible for movements and contractions, crawling, pinching, and formation of extracellular extensions such as pseudopods. Really heavily involved in phagocytosis and those sorts of things that change the shape of the cell membrane. Microtubules are long, hollow tubes, and they actually have things that flow inside of them. Help really with the shape of the eukaryote, because you remember eukaryotes, or at least the ones that don't have a cell wall, which would not include plants, of course, transport substances from one part of the cell to another, and these are what make up the spindle fibers, which, if you remember cell division during anaphase, these spindle fibers which are connected to the chromosomes, actually pull each chromosome to opposite ends so that the cell can divide and become two separate cells. We'll talk about that at some point. And then the last type are called intermediate filaments. They're called intermediate because they're halfway between the size of the microtubules and the actin filaments. About 10 nanometers in diameter. I'm not concerned that you know these sizes, by the way. Generally, I consider that to be trivia. I don't think it's the kind of knowledge that's useful or easily remembered, so it just bogs us down. So don't worry about that. Anyway, structural support to the cell organelles. So this is just a chart that it really doesn't say anything that we haven't already talked about. But you might take a minute. It kind of sort of puts everything that we already talked about in the last two chapters all into one chart. So if you want to take some time and kind of look over that on your own. Which of the following organelles is responsible for energy production in the cell? Nucleus, Golgi, ER, mitochondria, ribosome, and the answer to that would be the powerhouse of the cell, which is the mitochondria. So now we're going to talk about some specific types of organisms. So fungi or fungi, you can say it either way. I don't know what's right. You hear it both ways, so I don't think it really matters. Fungi sounds a lot more fun though. So approximately 100,000 species of fungi, broken up into two groups, which is they're either macroscopic, which means you can see them without with the naked eye. So mushrooms, puffballs, gill fungi, or microscopic fungi, which would be molds and yeasts. So fungi can have different forms. They can either be unicellular, which is one cell, colonial, which is where separate organisms live together, or multicellular, which is where they're all part of the same organism with specialized cells. Mushrooms and puffballs would be the example of that. So let's talk about the forms of the cells, starting with yeast. So yeasts are usually roundish or oval. They reproduce by something called asexual reproduction, which means that they don't need other yeasts to be able to reproduce. And they reproduce by a process called budding, just like SpongeBob SquarePants. The other form is the hyphal form. And so these are long thread-like cells that you see in filamentous fungi. When you see mold on a piece of bread and it looks like fur, that's what you're looking at. There are also these things called pseudohyphae. Pseudohyphae look like hyphae. That's what pseud means, false. But they're not. Instead of being a filament, they're actually chains of yeast cells that once you look closely, you can see that they're actually not a filament, but they are just a long chain of these yeast cells. So some of these have a life cycle that is dimorphic, which means they can either be hyphal or yeast form depending on what the conditions are in which they're living. And it's a common scenario that you see in some disease-causing molds. So this is just showing a graphical representation of yeasts, a yeast, and yeasts. That's a yeast with a bud coming off of it. Pseudohyphae. This right here is an electron microscope picture. And I may have mentioned this or not, but wherever you see colorful electron microscopic pictures, you got to understand that they color those with a computer. They're not actually, they don't even really know what color these things are. Or at least I shouldn't say that. What I should have said is they don't see color in an electron microscope. It's black and white. So let's talk a little bit about the pathogenicity of fungi. So they can make anybody sick. these primary pathogens can, no matter if you're healthy or not. Opportunistic pathogens are different. What they do is if you're already weakened because of something else, another health issue, then these opportunistic pathogens like yeasts and things like that. I mean, you've all heard of yeast infection and whatnot. Not everybody gets them, but some people do. Mycoses. So remember I told you that MYC means fungus. So this literally means a condition of fungus. But that's what we call fungal infections. Really can take on all kinds of different symptoms, um, depending on, you know, where the or how the thing infects a person, how healthy a person is. The way a fungal infection infects a person varies dramatically. It can enter in various different places. It can be foodborne. There's even, this was new to me, this last year I learned about uh, fungal meningitis, which, if you listen to the news, was from a contaminated injection of some sort. I think some kind of a pain injection. But a bunch of people died from it. So, anyway, all kinds of different ways that these things can cause people to be sick. All right, some other things. Harmless spores can cause infections in AIDS patients because AIDS patients, their immune system isn't working. Some fungal cells are an allergen to certain people, so it creates a harmful immune response, just basically an overactive immune response. Um, Mushrooms can produce poisons that can kill people. That's why you're not supposed to just go around eating mushrooms out of the woods. Um, if grain doesn't stay dry, there's a fungus called Aspergillus flavus that can kill animals who eat the grain that that it's growing on. Um, Generally, there are animals that are more sensitive. Horses, for example, are a lot more sensitive than cattle. Um, there are some fungi that will actually infect plants. Um, you have to All you have to do is go to the plant aisle of a nursery or something and see the things that they have, the sprays and whatnot that are for different kind of plants. Um, Fungi can also cause food to spoil. We've all seen mold on bread and mold on, things that you've left in the refrigerator for too long. That's fungus that you're looking at. Generally, if it forms like a slime, it's generally a bacterium that is growing. And if it's kind of that furry looking stuff, it's generally going to be a fungus. So they're not always bad, though, of course. I mean, we eat them, shiitake mushrooms and whatever, delicious. Uh, but the other things that they're beneficial for is they decompose organic matter. So when things die, I mean, if they didn't decompose, the earth would be covered in dead animals and dead plants and all kinds of stuff. So fungi are one of the things that are very important in decomposition of organic matter, which creates soils and all kinds of beneficial things. There are also associations that fungi have with plant roots, um, that help the plants actually absorb nutrients and water better so that they can grow better. We can also engineer fungi to produce antibiotics, alcohol, which is how we create beer using yeast, which is a good thing, organic acids, vitamins, things like that. And as I mentioned, some fungi we use to eat, like mushrooms, and I don't know anything else. Well, I guess yeast, you eat that, sort of, brewer's yeast and things like that. Um, so how about the way fungi live, the kind of things they eat? So there's this word called heterotrophic, and hetero means different or kind of opposite. Troph means develop or development. So heterotrophic uh, fungi get their nutrition from a wide variety of substances or substrates. Um, a saprobe gets its nutrients from dead things, dead animals, dead plants, things like that. Parasites that are parasitic fungi have to grow on an animal, but it's got to be alive. So a saprobe can grow on a dead animal. Parasites grow on live animals. Um, so there are not very many obligate parasitic fungi, meaning they can live on a live host, but they don't have to live on a live host. So the way that these fungi eat is they grow into the substrate, whatever the food is that they're living on, and they start secreting enzymes which break it down, and that actually digests it on the outside. So like a human, we digest things inside of our GI tract and then absorb it. Well, these things digest it on the outside and then absorb it. So some of these things can thrive in very nutritionally poor environments or really adverse environments where other things would have a very hard time living. Um, some of them can live in very saline environments or environments that have a very high sugar concentration, like honey. I mean, if you one of the old sort of old remedies is putting honey on wounds, and the reason why they do that is because it creates an osmotically poor environment for bacteria to live on, and so they essentially just dry out. It just sucks it does the same thing salt does. That's why you can put salt on, I don't know, meat like pork, for example. You've heard of salt pork or ham. Um, that's the reason why they do that is to, originally that's the reason why they did it was to preserve it because um, microbes can't live on it because the water will diffuse out of them and they'll die. Okay, so the morphology, and remember morph means form, so the study of form, or just really this just means the way they look. Um, so most fungi grow together, either loosely associated with each other or in tight-knit colonies. Colonies of yeasts act kind of like bacteria, look kind of like bacteria, soft, uniform texture and appearance. Those filamentous fungi, like I'd mentioned earlier, are cottony, hairy, velvety. So they look, the word I think I used was furry. They look like fur growing on stuff. So there are these things called mycelia. And just what that is just the mass of these filamentous fungi um, that, again, like I said, looks like fur. And so this is showing a mass of mycelia right here. Septa. So if you look at this picture right here on the left, you can see how this is all one continuous piece, but this one here is divided. A septum is nothing, it's just a wall. That's what the word means. And so some of these are segmented, and what separates the segments are a wall that we call septa. Um, so even though they're separated, they still have channels in them that allows things to be passed from one section to another. So non-septate hyphae, which is the one on the right there, the rhizopus, only have just one continuous long cell. So penicillin is septated and rhizopus is non-septate. So vegetative hyphae are responsible for what we see when we look at a mold, um, which is different from the spore because we also have these reproductive or fertile hyphae, which are what actually produces the spores. So how do these things reproduce? Um, so they reproduce by growing out these hyphae, and they can create spores. So the spores are what are responsible for reproduction, and these are similar to the endospores that we discussed with the prokaryotes. These things can produce spores by the billions, and that's what makes people sick a lot of times. Like for example, if you have mold in your home, they release these spores out through the air, and then when people breathe them, they get mycoses of the lungs. They, an infection, and they start producing poison because they're actually living in your lungs. But they can be released in water. They can be attached to other animals and so on. And so what happens is, just like with the bacteria, once conditions are favorable, they will germinate, so to speak, and produce a new fungus. And from one fungus grows 2 and 4 and 8 and 16 and so on until you've got, millions of cells again. So let's talk about how we make a spore. So as we mentioned earlier, they are asexually reproduced. So there's 2 types of this reproduction, or not reproduction, but spores. So the first one is a sporangiospore. So it's formed by successive cleavage, or cleavage is a word for splitting. So splits and then splits and then splits of this sac-like head called a sporangium, which is what we're looking at right here, which is attached to a stalk called the sporangiophore. That's Phore, 4. The other type is called a conidiospore, also known as a conidia. These are free spores that aren't enclosed in a sac. So they just kind of pop off of the fungus rather than being enclosed in a sac. Some of these things, the sporangiospores will actually um, pop and explode and go shooting, out into the air. So the other way, besides asexual though, is sexual spore formation. And the benefit to sexual spore formation is variation of genetics. And as I've mentioned before, variation of genetics allows, generally speaking, for better adaptation. And so, just like with other sexual reproduction, there's got to be 2 parents. And those parents will link their DNA to create offspring that are a combination of the 2. Potentially, I said that already. So conjugation methods, meaning how the 2 get together, so to speak, vary from just simple fusion of the cells to extending these fertile hyphae um, with female and male structures, almost like, gosh, do I dare say having sex? That's technically not a scientific term. It would be intercourse, I suppose. Anyway, but something along those lines. Okay, so which sources can fungi derive nutrients? Dead plants and animals? Yes. Saprobes. Living tissues? Yes. Rubber? You may not think so, but yes. Remember, rubber, by the way, is it's produced in plants, so probably isn't going to live on the rubber on your cars. That would be vulcanized rubber. That's a little different. Petroleum products, which, don't forget, those also are organic matter that comes out of the ground because of living animals, right? So it wouldn't be probably a stretch to imagine that it's all of the above, right? Well, that was fun. Fungi is cool to talk about. So now we're going to switch gears and talk about protozoa, which is plural for protozoan. If you stick an end on the end there, that would be singular. An N on the end. Did I say that right? Um, so the word protozoa comes from the Greek word for first animals, because at one point um, scientists thought that these represented the first animals. They don't, but that doesn't really matter. So there's about 65,000 species of single-celled organisms called protozoa. Most of them are harmless. They live in water and soil, like ponds and things like that. A few of them, however, are parasites and create millions of infections and some deaths every single year. So they're single-celled organisms that have all of the major organelles of any eukaryote, except they don't have chloroplasts. And one interesting feature about these protozoa is their cytoplasm is actually divided into two parts. The first part is called ectoplasm, and this is the outer layer that these organisms use for moving, feeding, protection. And the inner layer, so this is just inside of the cell membrane. Um, and then there's actually a deeper layer of cytoplasm, or not a layer because it's all the inside, and it's called endoplasm. So ecto and exo mean the same thing, which you probably know means outside, and endo means inside or within. So this is the granular inner region. When we say granular, I don't like that word very much, but it's not my decision. What it means is there's solid particles inside of the inside of the cytoplasm. That's all that means. We use that when we talk about white blood cells too, granulocytes and agranulocytes. It just means there's stuff in there. Um, so it's the inner part that has the nucleus, mitochondria. This is what we think of with when we think of cytoplasm. Okay. Um, so in protozoa, they have sort of a primitive nervous system, and they need that because protozoa move. We say they move in an amoeboid fashion because an amoeba is a protozoan, which, by the way, is what you're looking at right there. That is an amoeba. So let's talk about how they're shaped. So sometimes these things have a constant shape, oval, spherical, whatever. Most of the ones that have a constant shape are ciliates, meaning they have cilia on them, because if they have a constant shape, they're not going to be able to move unless they do have cilia. So the cilia act like oars so they can swim around. Um, the other type changes constantly, and these are the amoebas. You can see this picture on the right, how this thing sort of appears to be crawling, and that's exactly what they do. So they send out these little feet, and then they just sort of roll around, like a blob, if you've ever seen that movie from the 60s. Um, so again, different types of amoeba. Um, Heterotrophic, meaning they require food different. Heterotrophic again means different, right? So they require a food source from a wide variety of sources. So free living species can live on dead plant or animal debris, um, live off of bacteria by eating them, eat algae, and so on. Some species do have some feeding structures. One is called an oral groove. Oral means mouth. So it sort of acts like a mouth. And some protozoa can absorb the food just directly right through their cell membrane. Parasite protozoa have or live on the fluids in the host that they live on. So the main limiting factor for them growing is the availability of moisture. They pretty much all have to have a wet environment, but it can be anything from salt water to moist soil to plants inside of animals, even maybe on animals, and so forth. Some of them can survive in very extreme temperatures and very extreme pH levels. Some of these can create a cyst, and a cyst is really similar to a spore. Certainly, you can compare the two. So it's resistant, the dormant stage of the species, just like the spore was. So this picture right here shows this formation of a cyst. So the first phase of the life cycle of a cyst forming amoeba is called the trophozoite. So it is the stage in which it can move. This is the stage that has to have a lot of food, has to be moist, and once those conditions are no longer present, that's when we begin to create a cyst. So the cyst is the dormant stage that forms, as I said, when conditions aren't favorable. Now, it does not form a cyst the same way that a prokaryote forms an endospore. Remember, the prokaryote basically doubled its DNA and formed a sort of a smaller, more resistant copy of itself. Well, that is not what happens here. If you look at this picture here, you can see that all that happens is the trophozoite actually just changes shape and really reinforces its wall. But what it does is it makes it resistant to heat, drying, chemicals, pH, that sort of thing. Can be dispersed through the air, in the wind, um, by in the water, whatever. And this is very important in the spread of disease because it's more difficult to kill these cysts than it is to kill the trophozoite, right? So the trophozoite goes from being this sort of oddly shaped structure to a spherical shape. And the ectoplasm starts to secrete uh, this material that reinforces the cell membrane and makes it very tough and resistant, which is what this is showing right here, the cyst wall formation. So some protozoa groups only or exist only in the trophozoite phase, and others go back and forth from the trophozoite to the cyst stage, of course, depending on what the environment is, right? An example, Trichomonas vaginalis is an STD that causes trichomoniasis. Doesn't form cysts, and since it's an STD, obviously needs to be transmitted during intimacy, right? Entamoeba histolytica and Giardia lamblia both form cysts. You've probably heard of Giardia. This is why we don't drink water um, straight out of the stream, especially in high altitudes, because this Giardia lives up there. Anyway, transmitted, of course, by contaminated food or water. So all protozoa reproduce by asexual mitotic cell division or multiple fission, which is just a different type of division. Some, however, can also reproduce by sexual reproduction, where they conjugate nuclear material. Ciliates are one that participates in this conjugation. And what happens is the two cells fuse and they exchange their nuclei. And as we've mentioned before, this allows for more genetic variation, which can potentially make them more adaptable to their environment. This table here just shows some kind of common protozoa and the diseases that they cause and where they come from. So take some time and look over that on your own. The active feeding and motile stage of the protozoan life cycle is the cyst, endospore, trophozoite, merozoite, or all of the above. And of course the answer is trophozoite. All right, helminths. Sounds like helmet, but it's not. Um, the word helminth comes from the Greek word for worm, and you can imagine what these things are. They're worms. And it includes tapeworms, flukes, and roundworms. Some of these things are so big that you can see them with the naked eye. Some of them can be several meters in length, but others of them are microscopic. Not all of these are parasites. Some of them just live in can live in soil or water, and some of them can be either. Parasitic helminths, though, spend part of their lives in the GI tract of animals. So there's two major groups of these helminths: flatworms, which is the phylum Platyhelminthes. I believe plat actually means flat. I'm fairly certain of that. These can also be called nematodes. And if you know SpongeBob, that's a bad infection. They have thin, often segmented bodies So they're separated into sections like this picture is showing over here. And they are divided into either cestodes, which are tapeworms, or nematodes, which are flukes, like a liver fluke. Yeah. So flatworms is the first major group of helminths. The other major group are the roundworms, and they are of the phylum Aschelminthes. So they have an elongated, cylindrical, unsegmented body. So they look kind of like just a typical disgusting worm. But let's talk about the morphology of them. So they're multicellular organisms, which means they've got organs and organ systems, just like every other multicellular organism has. The most developed organs are the reproductive tract because that is their main goal in life, is to reproduce. They do have digestive, excretory nervous system and muscular systems, but they aren't as developed as the reproductive systems. Okay, so there's 3 stages of the lifestyle of these helminths. Starts with a fertilized egg, also known as an embryo. Once that embryo hatches, or the egg hatches, they go into a larval stage. So it's a larva. The larva then continues to develop and eventually becomes the adult. So adults derive nutrients and reproduce sexually in the body of a host. Nematodes have different sexes, males and females, and they don't look the same. Trematodes may have separate sexes, male and females, or they may be hermaphroditic. A hermaphrodite is both male and female. Cestodes are generally hermaphroditic. So the life cycle of a helminth, first of all, it's got to transmit some sort of an infection, either an egg or a larva, to a host. And the host that develops the larva is known as the intermediate or secondary host. Okay, now that isn't necessarily the same organism that the adult lives in. Okay, the adult lives in what is called the definitive or the final host. So examples of the way humans are infected could be with contaminated food, soil, water, or infected animals. Um, Probably, I think the most common route of infection is what we call fecal oral, which means fecal contamination in our food. So from the feces of one animal to the mouth of another animal. Lots of diseases are spread that way. That's why you need to wash your hands. Another way they can penetrate though is through skin, and it doesn't have to be a cut. They can just bore their way through the skin. It's fairly disgusting. So this table just shows again some common types of helminths, the disease they cause, um, how they're spread, and so on. So we're going to cover, as I've told you before, most of the pathology in your pathology class. So, but you should really take some time to kind of just look over this for a little while. One that's interesting that you may want to be aware of is this Tiniosolium, which is the pork tapeworm. This is why we need to cook pork right there. So back to this life cycle. I shouldn't have put that table where I did, but anyway, these fertilized eggs need to be released into the environment, right? What they do, they provide a sort of extra protection from the environment as well as food. It's like a chicken egg. all of the food that that chick needs to develop is inside of that egg. And that's the sort of the same thing. So they have the ability to develop into the larva before they hatch. Now, in this stage, they're vulnerable to heat, cold, drying, and predators, and so meaning things that actually eat them, they eat the eggs. So they have to survive this stage, obviously, before they can become larval. So certain helminths can lay 200,000 eggs a day, because the more eggs they lay, the greater chance they're going to have of having some sort of viable offspring. Some of you have maybe had this or seen kids with this or seen dogs with this. If you ever see a dog dragging its rear end on the floor, pulling itself along with its front legs, it's most likely got pinworms because they tickle and itch the anus. So the pinworm is Enterobius vermicularis, also called a seat worm, I suppose because it affects your seat, or you take a seat. I don't know. Very commonly a common infection, and what it does is they grow in the large intestine, which is your colon, right? 2 to 12 centimeters in length, and they have a curved or cylindrical shape. Now, just so you know, it's not these things that are coming out of the anus. There's not a 12 centimeter long worm crawling out that makes it itch. It's actually the eggs. And you may, I don't know if you've ever thought about this, but the way viruses spread, one of the ways is by causing a person to cough. And that then, of course, spews the virus out into the air where it can be picked up by someone else. So these pathogens can trigger these undesirable sensations in a human to sort of help them spread themselves. Well, what these things do is as these eggs come out, it causes the anus to itch. And so little kids scratch their rear end, and then they go play with other little kids and transfer those eggs to the other person. And these can actually come from dogs too, by the way. So if you have a dog that's got an infection, it can easily spread to a human. So the life cycle of a pinworm is just what this picture shows. You've got to swallow it, so it's a fecal oral infection. And of course, then it begins to grow after the eggs hatch. After about a month, the larvae become adults, and then they begin laying eggs. After they mate, the female migrates to the anus and she lays her eggs, and then it causes itching, and of course, just like the picture shows, scratching and spread to another person. Okay, so there are about 50 species of helminths that will become parasitic in humans all over the world. There are higher incidence in tropical areas because the worms can survive outside of a host in these areas much more easily than they can in areas like with more extreme environments, extreme heat and extreme cold, which would be, away from the equator primarily. So there are billions of infections all over the world, all different classes of people and socioeconomic status and whatever. Anyway, estimated that at least 50 million infections in North America alone every single year. So adulthood and mating of the helminths occurs in larvae stage, in the intermediate host, as a cyst, or in the definitive host, or in the egg stage. And of course, the answer is in the final or definitive host. And that is chapter 4.